Welcome to Lab 3, Electrophoresis. Before you even get into the lab, we're going to use these materials to create the buffer solution and agar gel that you will use to run your gels. This includes a 1x buffer and agarose powder. Here you see us portioning out the buffer for use in the agarose gel. Once the buffer is diluted, we can measure out 0.75 grams of agarose powder to add to the buffer. This buffer will now be placed in the microwave to heat it up to a boiling point. Make sure that you never use the microwaves for anything other than lab-authorized experiments, certainly not for food and drink. Always use gloves when handling hot materials. Once our solution has cooled down so as not to be hot to the touch, we will add it into our gel trays. Your gel trays come in two different sizes, large and small wells. For this experiment, we'll be using large well sizes, but in the future, we'll be using small ones. Here you see Gio holding a gel tray. You should never be removing your gel from its tray without explicit instructions to. Now that we've poured the gels, they'll take about half an hour to set before they can be used. The gels that we make can sometimes slide out of their trays, so make sure that you hold both the gel and the tray when transporting it. When you come into the room, you will likely see a gel electrophoresis chamber with a gel already in it, as well as buffer already around the gel. If you have to add the buffer yourself, be careful to pour it evenly across both sides so as not to upset the orientation of the gel already in the container. The liquid buffer should cover your gel pan, so make sure that there is more liquid over the gel itself. For this experiment, you will need your 0.5 to 10 microliter pipette, as well as the provided U-view and ladder that should be available in your kit when you walk in. In addition to your pipette, you should have your 4 tubes from last class and an additional tube in which you'll place your ladder. Once your pipette is set to 3 microliters, you'll pipette ladder into your ladder designated tube. It's okay if it doesn't look like that much liquid. It really isn't. Before completing this step of the process, ask your TA whether 3 microliters is enough. Different ladders require different amounts, and sometimes our ladders change. After our ladders, we're going to add our U-view dye. 4 ULs of UV dye should be added to each of your small microtubules. During this process, make sure that you replace your tip after every single tube, as you are using your pipette to mix as well as add. We're now going to add 6 microliters of our ladder to our gel. Make sure that your pipette tip is placed all the way into the well, but does not poke into the gel itself. You may want to use your finger to stabilize, as Geo does in this video. After the ladder, we will add 7 microliters of our positive control, negative control, fish specimen 1, and fish specimen 2. These should have been prepared last class. This video is sped up, so be aware that the piping of the gels might take you a substantial amount of time. Once your gels are piped, they should all be visible from above and from the side. Be aware that your gel should not be multiple colors as these are. This is because it turns out we were using expired products. We are now adding the lid to our electrophoresis chamber, making sure to line up the red with the red wire and the black with the black wire. The wells on the gel should be closer to the black side, and remember, always run to red. We will now set our electrophoresis chamber for 120 volts for 30 minutes. Make sure that your chamber is running by checking for bubbles on both of the electrodes. The red side is our positive electrode, and the brown side is our negative electrode. Once our run is done, we will carefully unscrew our wires, making sure not to tip the chamber in the process. Because of our poor materials, the gel that we ran here did not turn out very well. Even though the materials for this gel are bad, we're going to show you what happens now. We go into the second adjoining room in the lab, put our gel onto the UV viewing table, slide it off of its gel shell, and then turn on the machine to view. Make sure that your lights are off, or else you won't be able to see what is on your gel. Click to the next image to see what a proper gel run for this lab looks like.